हेलो फ्रेंड्स, आज सेल साइकिल एंड सेल डिवीज़न का पार्ट 2 पढ़ेंगे और आज चैप्टर का एंड हो जाएगा। साइटोकाइनेसिस माइटोसिस accomplishes not only the segregation of duplicated chromosome into daughter nuclei karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm and called cytokinesis, at the end of which cell division gets completed. In animal, cytokinesis occurs by constriction and furrow formation. Microtubules and microfilaments arrange on equator to form mid-body, and at the periphery of the equator, a contractile ring is formed that is made up of actin and myosin protein. Due to interaction between actin and myosin, ring contract. Thus, a furrow forms from outside to inside in cell. Furrow depends continuously and ultimately a cell divides into two daughter cells. In animal, cytokinesis occurs in centripetal order. Cytokinesis in plants takes place by cell plate formation because contraction is not possible due to presence of the rigid cell wall. Many Golgi vesicles and spindle microtubules arrange themselves on equator to form phragmoplast. Fragments of endoplasmic reticulum may also deposit in phragmoplast. Membrane of Golgi vesicle fuse to form a plate-like structure called cell plate. Golgi vesicle secrete calcium and magnesium pectate. Further, cell plate is modified into mid-lamella. In plants, cytokinesis occurs in centrifugal order. Cell plate formation is formed center to periphery. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arises, leading to the formation of syncytium. For example, liquid endosperm in coconut. Significance of mitosis. Development of an organism occur by mitosis. Every organism starts its life from a single cell, for example, zygote. Repeated mitosis in zygote leads to the formation of the whole body. The growth of multicellular organisms is due to mitosis. Number third, cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. Number 4, a very significant contribution of mitosis is cell repair. The cell of the upper layer of the epidermis, cell of the lining of the gut, and blood cells are being constantly replaced. Number 5, mitotic division in the meristematic tissues, the apical and the lateral cambium result in a continuous growth of plants throughout their life. Modification of mitosis. Number first, free nuclear division karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arises. Number second, endomitosis. This is duplication of chromosomes without division of nucleus. Endomitosis leads to polyploidy. For example, increase in number of set of chromosomes. Colchicine induces polyploidy in plants. Colchicine is a mitotic poison as it arrests the formation of spindle fibers. Number third, endoreduplication. Endoreduplication is a modification of endomitosis. The polytene chromosomes are formed by the process of endoreduplication. In endoreduplication, the chromatids replicate but do not get separated. This process is also known as polyteny. Note: amitosis. It is a simple method of cell division which is also called direct cell division. In this division, there is no differentiation of chromosomes and spindle. The nuclear envelope does not degenerate. The nucleus elongates and constricts in the middle to form two daughter nuclei. This is followed by a centripetal constriction of the cytoplasm to form two daughter cells, for example, prokaryotes and some unicellular eukaryotes. Meiosis. Term meiosis was proposed by Farmer and Murray. The specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, where fertilization restores the diploid phase. Meiosis occurs during gametogenesis, leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis first and meiosis second, but only a single cycle of DNA replication. Meiosis first, heterotypic division or reduction division. It leads to reduction in chromosome numbers. Division of chromosome does not occur in meiosis first. Only segregation of homologous chromosomes takes place. Meiosis first is initiated after the parental chromatids have replicated to produce identical sister chromatids at the S phase. Meiosis first involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. Meiosis second. This is a homotypic division of equational division. It does not lead to any change in chromosome number. Division of chromosome or centromere occurs during meiosis second. Four haploid cells are formed at the end of meiosis second. All the four daughter cells produced by meiosis are genetically different from each other and also differ from the mother cells. In meiosis, division of nucleus takes place twice, but division of chromosomes occurs only once in meiosis second. Meiotic events can be grouped under the following phase. First meiosis first, then meiosis second. Under meiosis first, prophase first, metaphase first, anaphase first, telophase first, and then under meiosis second, prophase second, metaphase second, anaphase second, and telophase second, then interphase. Same as in mitosis. Stage of meiosis. Prophase first, typically longer and more complex when compared to prophase of mitosis. Prophase first is classified in 5 sub-stages based on chromosomal behavior. First leptotin, then zygotin, then pakitin, then diplotin, then dikinesis. First duplicated chromosome. In zygotin, synapses form. In pakitin, crossing over occurs. In diplotin, synaptomial complex and disappearing of and chiasmata visible. Then last stage, dikinesis, bivalent, ready for metaphase. Number first, leptotin. Chromatin threads condense to form chromosome. Chromosomes are longest and thinnest chromosomes become gradually visible under the light microscope. All the chromosomes in nucleus remain directed towards centriole. So group of chromosomes in nucleus appears like a bucket stage. Number B, zygotin or synaptotin. Zygotin is characterized by pairing of homologous chromosomes. Synapses, pairs of homologous chromosomes are called bivalents or tetrads. However, these are more clearly visible at next stage, pakitin, a structure developed in between homologous chromosomes which is termed as synaptomial complex. The first two stages of prophase first is relatively short-lived compared to the pakitin. Pakitin thick thread. During this stage, the four chromatids of each bivalent chromosome become distinct and clearly appeared as tetrad. Recombination nodules between non-sister chromatids of homologous pair developed and these non-sister chromatids exchange their parts, for example, crossing over. Crossing over leads to recombination of genetic material on the two chromosomes. Crossing over is an enzyme-mediated process and the enzyme involved is called recombinase, endonuclease plus ligase. Recombination between homologous chromosomes is completed by the end of packaging, leaving the chromosomes linked at the sites of crossing over. Number D. Diplotin. The beginning of diplotin is recognized by dissolution of synaptominal complex. Homologous chromosomes start repulsing each other, so X-cell structure appeared called chiasmata. Diplotin may last long up to months or years in oocytes of some vertebrates. Dicytotine. And then number E. Dikinesis. It is final stage of meiotic prophase, first marked by terminalization of chiasmata. Chiasmata open in zip-like manner. Chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosome for separation. Centrioles move towards the opposite poles. By the end of dikinesis, nucleus disappear and the nuclear envelope also break down. Dikinesis represents transition to metaphase. Number 2. Metaphase first. Bivalents arrange on equator congregation of cell to form metaphase plate. The microtubules spindle fiber from the opposite poles of the spindle attached to the pair of homologous chromosomes with one kinetochore of each chromosome. Two types of spindle fibers appear in the cells. Number first, chromosomal या kinetochore spindle fibers. Number second, supporting या continuous या non kinetochore spindle fibers. Number third, anaphase first. Due to sorting of kinetochore या chromosomal fiber, homologous chromosomes segregate from each other and moves towards the opposite pole. Sister chromatids remain associated at their centromeres. For example, chromosomes remaining in double chromatid stage. Anaphase first is characterized by segregation or disjunction of chromosomes. Division of centromere is absent. Number 4. Telophase first. The nuclear membrane and nucleolus reappear and cytokinesis occur along with telophase first to produce two haploid and daughter cells. This is called as dyad of cells. Although in many cases of the chromosomes do undergo some dispersions, but they do not reach the extremely extended state of the interphase nucleus. Prophase first, metaphase first, anaphase first, then telophase first. Interkinesis. Gap between meiosis first and meiosis second is called interkinesis. Preparation of meiosis second occur during interkinesis. It is like interphase of mitosis, but replication of DNA is absent in interkinesis. Interkinesis is generally short-lived. Interkinesis is followed by prophase second, a much simpler prophase than prophase first. Stage of meiosis 2. Prophase 2. Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis first and meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. The chromosome again become compact. Metaphase 2. At this stage, the chromosome allied at the equator and the microtubule from opposite poles of the spindle get attached to the kinetochore of sister chromatids. Number 3. Anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosome which was holding the sister chromatids together, allowing them to move toward opposite poles of the cell by sorting of microtubules attached to kinetochores. Number 4. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, for example, 4 haploid daughter cells. prophase 2, metaphase 2, anaphase 2, and telophase 2. Significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generations in sexually reproducing organisms. Even although the process per paradoxically results in reduction of chromosome number by half. Number two, it also increases the genetically variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution.